Hello everyone, this is All Ali, and now we will discuss the criteria of action potentials. The first thing is that action potentials are all or none, either to happen completely or not to happen at all. So if the stimulus is strong enough to get the membrane to the threshold, which is minus 55 millivolt, in this case, the action potential happens. If the stimulus is weak and the membrane is not able to go to the threshold, which is minus 55 millivolt, action potential will not happen. When it happens, it happens completely unless it goes to up to the peak, which is plus 30 millivolt. So all action potentials are the same. All of them, they have a complete response. They go up to the peak of plus 30 millivolt. The second thing is that action potentials have refractory periods. There are two types of the refractory periods, the absolute refractory period and the relative refractory period. In the absolute refractory period, in this time, another action potential cannot happen, absolutely cannot be initiated, no matter how large a stimulus is applied. Why is that? Because when the peak is achieved, which is plus 30 millivolt, the voltage gated sodium channels are inactivated and they cannot be activated again until the membrane goes down to be hyperpolarized. So during this time, which is the absolute refractory period, the action potential cannot be initiated again due to the inactivation of the sodium voltage gated channels. Then the relative refractory periods happen during the time of hyperpolarization. They are relative because they need stronger stimulus than usual to be able to initiate the action potential. Why? If you start a stimulus at the resting state, which is minus 70 millivolt, the stimulus is needed to go up, to get the membrane up to the threshold of minus 55 millivolt. So the difference here is only 15 millivolt. The stimulus must be strong enough to go up to minus 55, so it needs to push the membrane up with 15 millivolt. But during the hyperpolarization, the membrane here is, say, minus 90 millivolt. And to go up to the threshold, a stimulus must be strong enough to push the membrane up from minus 90 to minus 55. So it has to be 35 millivolt. So it has to push the membrane up 35 millivolts. So it's able to go up to the threshold. So this is a relative because it needs really stronger stimulus that is able to take the membrane from minus 90 to minus 55. Action potentials are propagated once the threshold is reached. The action potential begins at the axon hillock and then it propagates down the whole length of the axon. When it propagates, it's not conducted decrementally, meaning it has the same strength. It does not becomes weaker over distance. And it is unidirectional. It goes only down the axon in one way and does not go to the opposite direction. Action potentials can be either continuous or saltatory. Continuous if the axon is not myelinated, and saltatory if the axon is myelinated. So in this case, action potential has to jump between the nodes of Ranvier, and it's going to be faster than in case of continuous conduction. So here is where action potential is initiated at the axon hillock, then it propagates unidirectionally down to the length of the axon. And when it propagates, it has the same strength over the axon. It always goes up to the peak of plus 30 millivolt. There are two types of conduction: continuous conduction if the axons are non-myelinated, or saltatory in case of myelination. So what is the difference here? If you look at here, the axons are non-myelinated. Here, the axons have myelin sheath. So at the very beginning of time, we have the action potential that is initiated at the axon hillock. When you go to this time of 5 milliseconds in continuous conduction, here is the action potential. In case of myelination, here it is at this point of the axon. At 10 milliseconds, the continuous conduction action potential is still here, but in case of myelination, it almost now at the end of the axon. This means that saltatory conduction is faster because the action potential does not go through the myelin sheath, so it has to jump between the nodes of Ranvier. What are the two factors that increase the speed of an action potential? Of course, myelination. Saltatory conduction is faster than continuous conduction. The second factor to increase the speed of the action potential is the size of the neuron. If the neuron has a larger size, it is faster when it comes to the action potential. So here, Z neurons are faster than Y are faster than X. Because again, the larger the diameter of the neuron, the faster the conduction. Local anesthetics, like the ones dentists do to do tooth procedures, they inject something to numb the teeth. This is called local anesthetics, like Novocaine and Xylocaine. Those ones block the voltage-gated sodium channels, and so they prevent action potential from happening, so we do not feel the pain during the procedure. So what happens is the membrane can go up to the threshold because here we have ligand-gated channels. They open in response to the neurotransmitters. So you can go up to this threshold, which is minus 55. At this point, nothing would happen because the medication here block the voltage-gated sodium channels, so depolarization will not happen, and so you will not feel the pain during the procedure. Now, we need to compare action potential to the graded potential. But before that, let's go over some review questions related to the action potential. So the electrical impulses gather and accumulate in what part of the neuron to initiate the action potential? They accumulate at the axon hillock. So action potential begins at the axon hillock. The action potential is all or none. They are non-decremental, meaning they do not become weaker over distance. And the spike phase of the action potential, when the membrane goes up to plus 30, which is the depolarization part of the action potential, this is due to the opening of voltage-gated sodium channels. Then the repolarization and hyperpolarization are due to the activity of voltage-gated potassium channels. Where does action potential happen? Action potentials begin at the axon hallux and then they continue at the axon of the neurons. What refractory period is caused by the inactivation of the voltage-gated sodium channels? This is the absolute refractory period. Local anesthetics block what kind of channels? They block the voltage-gated sodium channels so that they prevent the depolarization phase of the action potential. Now with graded potential. So remember that action potentials begin here at the axon hallux and then they continue down the axon. On the other hand, graded potential happen at the dendrites and cell body of the neuron. Graded potentials can be either excitatory when the membrane goes positive, so it is likely to go up to the threshold of minus 55. So here action potential is likely to happen. And they are excitatory in case of positive ions enter the cell, like sodium enters. Action potential can be inhibitory when they do hyperpolarization. So this happens in case of entry of negative ions inside, like chloride. When it enters, the membrane will go negative, and this is hyperpolarization. So hyperpolarizing graded potentials are inhibitory because they inhibit action potential from being initiated. Of course, if you compare that to action potentials are always excitatory. You know that neurotransmitters are released in the synapse and they open ligand-gated channels, leading to movement of ions. The graded potentials can be depolarizing, so the membrane goes positive, and when the threshold is reached, action potential will happen. So we call them the depolarizing graded potential or threshold potentials. They can be inhibitory if the negative ions enter or potassium exit, which is positive. So the membrane will go more negative, hyperpolarization, and they are inhibitory because they will prevent the initiation of action potential. So those neurotransmitters in the synapse can be excitatory when they lead to opening of sodium ligand-gated channels or calcium, so positive ions go inside the cell, like glutamine. It is an excitatory neurotransmitter. They can be inhibitory when they open potassium ligand-gated channels, so potassium goes out of the cell, or when they open chloride ligand-gated channels, so chloride, which is negative, enter the cell. So what will happen here is hyperpolarization and action potential will not happen. An example of an inhibitory neurotransmitter is GABA. GABA is inhibitory neurotransmitter. So it prevents action potentials from happening. Some neurotransmitters can be excitatory and inhibitory, depends on what receptors they stimulate, like acetylcholine, for example. How the neurotransmitter is released in the synapse. Remember that once the action potential goes down the axon terminals, voltage-gated calcium channels open, calcium go inside the terminals of the axon, and then they kick out the neurotransmitter here in the synapse. Then the neurotransmitter will work on ligand-gated channels found in the dendrites and in the cell body. Again, graded potentials happen at the dendrites and the cell body of the neuron. So what happens when you block those calcium channels? Calcium will not be able to enter, then the neurotransmitter will not be able to exit in the synapse, and the action of the neurotransmitter is going to be inhibited. Graded potentials can be summed. They can add to one another. Action potentials cannot. So this is an important difference between graded and action potentials. There are two types of summation. Temporal summation. So temporal from time. So here we have only one presynaptic neuron working, but instead of working slow, it's going to be working faster. So instead of doing, it's going to be working faster. And in this case, we call that temporal summation. Like, for example, when you are working yourself to shovel the snow, and instead of working slowly, you are working faster over time. The other type of summation is a spatial summation, spatial from space. So here we have a space occupied by many presynaptic neurons. They are working at the same time. So if you are yourself, your spouse, your child, the three of you are working together to fill out the hole or to shovel the snow. So that is the work of the three of you is going to be added to one another. We call that spatial summation. So here in temporal summation, we have only one presynaptic neuron working. In the two first scenarios here, the work is kind of slow, so the threshold is not achieved. But when they work faster, the threshold is reached and then action potential happen. In a spatial summation, we have here multiple presynaptic neurons. Here we have three working at the same time. So when A is working alone, action potential will not happen. B alone is not able to initiate the action potential. C alone also is not going to be successful to initiate the action potential. But when you add the three at the same time, working at the same time, now action potential can happen due to that spatial summation. So the graded potentials can be excitatory, and here action potential is likely to happen, or hyperpolarizing inhibitory action potential is less likely to happen. The graded potential size is proportional to the size of the stimulus. So remember that action potentials are all or none. When they happen completely with the same strength. They go up to the peak of plus 30. In case of graded potential, they can be weak or strong. So they are the rule of all or none does not apply to graded potential. Graded potentials become weaker over distance. Why is that? Because the dendrites and cell body do not have myelin sheath. Remember, myelin is the conduction. But because the dendrites and the cell body do not have myelin, as the graded potential goes down them, it becomes weaker. Action potential does not become weaker over distance because the axons are myelinated. So let's compare the graded to action potentials. In case of graded potential, there is no threshold. In case of action potential, you must go up to the threshold so that action potential can be initiated by opening up the voltage-gated sodium channels. Graded potentials can be summed by temporal summation or spatial summation. Action potential cannot be summed. Graded potentials happen at the dendrites and the cell body. Action potentials begin at the axon hillock and then they continue down the axon. Graded potentials do not have refractory periods. Action potentials have absolute and relative refractory periods. The graded potentials happen due to the activity of ligand-gated channels that respond to the neurotransmitter or the ligand. Action potentials happen due to the activity of voltage-gated channels. All or none rule only applies to action potentials. Graded potentials vary in their duration, but action potentials have the same duration for a given cell type. So if the cell has the same diameter as others, they will have the same duration. Of course, if the cell has a larger diameter, the action potential is going to be faster. But here I'm referring to the same size cells. They have the same duration of action potentials. Graded potentials can be excitatory or inhibitory, but action potentials are always excitatory. So let's go over that review question. So graded potentials have no refractory periods. Graded potentials can be summed spatially or by temporal summation. Graded potentials are what? Because the dendrites and cell bodies are demyelinated, they are decremental, meaning they become weaker over distance. Where do graded potential happen? They occur at the dendrites and cell body of the neuron.